As you're aware, if you have been following these webinars, there are a few technical things to go over briefly. We have muted all of your microphones. If you want to ask questions while I'm talking, please use the dedicated Slack channels, and my colleagues Emily and Aaron will answer to you. All materials for this course will be available on this website mentioned here. We will update the videos with today's recordings, as well as the exercises and the solutions that you can find in the menu on the left. You can get help with these exercises by referring directly to the solutions, by using the Slack channels again, and also by emailing us at helpdesk@ensembl.org. So now I'm going to be talking about uploading your data to Ensembl, which is the seventh part of our webinar series, and later Emily will follow on with introducing the Ensembl REST API. This session will be structured in a similar way as previous sessions. So we're going to have a presentation which is split into two parts. In the first part, I will talk about visualizing your own data, and it has an associated demo with it where we will upload a bed file. You can find this bed file in the Slack channels where I uploaded it. We have a second part of the presentation where I will introduce track hubs, and then we have a demo associated with that as well. And as before, the exercises will be available online. Okay, so how do you visualize your own data? If you have followed this webinar series, you may have seen this custom tracks button on various pages before. This is a way how you can upload your own data. Your data has to be in one of several file formats that we support, and these range from bed files. This is a column-separated file with just coordinates and features on your genome. We will use that in a minute. You can have GFF files. You can have BEM or CRAM files with next-generation sequencing reads, and you can also have variant calling format files, as well as some other not so commonly used formats. There's documentation on our website available on both how you upload your data using custom tracks, as well as on the formats shown in these links at the bottom. And the pictures here are just some examples of what you can do. So here on the left-hand side, you have a BLAST result, which is visualized underneath a gene. And here at the bottom, you have next-generation sequencing reads along the genome. So how do you add your custom track? In principle, there are two ways of uploading files. First and foremost, if you have a relatively small file, up to 20 MB, you can upload it from your own computer, or you can also upload it from a location on the web using a URL. However, if your file is big, such as a BEM file, for example, you will need to attach it via a URL. And you also need, in this case, an indexed BEM file in the same directory. Please be aware that if you in particular use these large files, that the file will be read each time you visit a page where this track is shown. So this can slow down Ensembl significantly, and because of that, think very carefully about whether you really need this track, and also if you have worked with it and then don't need it anymore, please delete it again. And I will show how that works in a minute. You can also create an Ensembl account, and if you have an account, this enables you to save your tracks and share them with others. And finally, just to say that you can use this, of course, but be aware that we only have a relatively trivial security. So do not use this functionality of adding custom tracks for any data that is sensitive, such as data that contains patient identifiers. Okay, so now I'm going to demo how you can do this. And for the demo, we will assume that we have 3 patients who presented with microcephaly and developmental delay. And these patients had large deletions on chromosome 5. What we're going to do is we will format that file. I've prepared the file before, and we will upload it to Ensembl. And this is the first introduction to the BET file format, here shown in red. So a BET file format is a column format, which has at least 3 columns. The first one being the chromosome, the second being the start position on the chromosome, and the third being the end position on the chromosome. And then you can have additional 9 columns, including one with a heading name. So here we use P1, 2, and 3 for patient 1, 2, and 3. So this is a format where you can just annotate features based on just chromosome coordinates. Okay, so I will leave my presentation now, and I will go to Ensembl and have a look how we do this. You can do this from various pages in Ensembl, but let's have a look at the documentation first. So I will scroll to the bottom of the homepage and use this link, use my own data in Ensembl. I will click on that. And here I've got my documentation about how to upload files. There's a custom tracks button here where you can directly access the interface where you can upload the file. But what I will do first is to go to the different file types, which are linked in the first sentence here, to see this list of file formats we support. And we do have very specific instructions of how you need to prepare your files and how the files need to look like. Because you will be surprised that while there should be a common format, not everybody defines the format in the same way. So I'll go to BET format now by following the first link. And here you can see what I just said in the presentation. We have 3 fields, chromosome start and end position, which are necessary, and you have more optional fields that you can add in the columns. So if you work with any type of file, please have a look at this documentation. I've made this file with the 3 patients, and it's on my desktop. So I've got this text file here, which is called p1-3.bet. And if I look at that file in a text editor, it looks exactly as I described earlier. Okay, so now let's upload this file. One way, as I said, you can just go back in the documentation where we have been earlier and use this custom tracks button. But you are more likely to be actually somewhere in the genome browser when you want to upload a file. So what I will do now is I will go to a region of the human genome. I will just click on our example gene BRCA2 here, and then follow the link to the location tab here in green on chromosome 13. And I will use the custom tracks button on the left-hand side that some of you might have noticed in previous webinars already. So I click on custom tracks to add my data here, which brings me to this interface with personal data. So I need to give my data a name. So if I call it P1-3, and it tells me I'm looking at the human genome assembly GRCH38. And you'll have 2 options now. So first and foremost, you could paste your data into this box here. If I do this from my file, you see that I now need to choose a format here from the drop-down menu. This is because if I paste in data, Ensembl is not able to recognize the format automatically. So I would just say bed here. The other option is, if I delete this, I can also go to choose file and just select my file. And now you see the format is recognized automatically as bed format. And then I just need to say add data. Click at the bottom button at the bottom. And this interface now tells me that my file uploaded successfully and that my nearest region with data is on chromosome 5. So I follow this link to chromosome 5, which opens the location tab on chromosome 5 and the region in detail view. And if I now scroll to the bottom of the page, I see that Ensembl is still loading my additional track. Now it's done. So we have got this track highlighted in yellow here, which is called P1-3, with my 3 deletions visualized in brown blocks. Yeah, as I said, once you've done this, especially with larger files, it's advisable, if you don't need the track anymore, to delete it again. To do this, you can also go again to configure this page. And then you have to use the personal data tab on the right-hand side. I click on that. And here you can see the custom track that you show at the moment. And to delete it, you just use the trash bin icon on the right. And if I close this window now, my custom track will have disappeared. Okay, I'm not going to wait for this. Oh, here we go. So it's gone now. There's one other place where you can upload your file, and this is on the species homepage. So if you, for example, go to the human species homepage by clicking on human at the top left here, there is this link here, display your data in Ensembl. And if you click on that, that brings you to the same interface that we have seen previously. Okay, I'll go back to the homepage because this was the end of the first demo. And I will go to my presentation again to talk about track hubs. Okay, so track hubs are a way for you to visualize detailed data directly from a variety of data sources. There is a collection of publicly available track hubs, which is called the Track Hub Registry, and you can find this under this link. And this contains more than 4,000 hubs for 135 species and 163 different assemblies. And this is a place where many large projects, such as ENCODE and Blueprint, you have heard about during the regulatory build webinar. This is where they deposit their raw data. So these projects create a lot of data for a lot of cell lines, and we as an Ensembl are not able to show all of this. So this is a place where that data is deposited, and you can then import it from the Track Hub Registry and visualize it in Ensembl by selecting certain cell types, for example. So this Track Hub Registry, as I said, you can use to find and then use different track hubs with different types of data, not only regulatory data, there are lots of other types of data as well, and view that in Ensembl. But you can also theoretically upload your own track hub with your data to this registry to be able to share it with other people, including your collaborators. So essentially what a track hub is, it is a collection of a set of tracks that is associated with a project or an experiment or something like that. These are some examples from the track hub registry and the track hubs that are available there. If you are interested in genome annotation and newest updates to annotations, or maybe looking at a gene that looks something a little bit dodgy, you may be interested in the GRC genome issues under review track hub. So this visualizes known problems with the human genome assembly. There's also one on gen count annotations. These are daily updates to gene annotations that you will otherwise not see in Ensembl, because we work on a 3-month release cycle. There are track hubs for non-coding RNAs, for proteins, for GTX data, and also for the different projects generating regulatory data, such as Blueprint, ENCODE, and Roadmap Epigenomics. You will have heard about if you watched our webinar on the regulatory build. So what we're actually going to do now is to add the Blueprint track hub, which contains epigenomics data, to Ensembl, and we will view a number of different tracks from this project. I'm going to leave this presentation again and say at this stage that there are 2 different ways of getting a track hub into Ensembl. One is to search a website that is called Track Hub Registry and import the data from there. And the second is to search for track hub within Ensembl and import the data. So what I will do first is to go to the Track Hub registry. So this is found under trackhubregistry.org. This is the website. Here at the bottom right, you have the stats with the different hubs, species, and Ensembl and assemblies that I mentioned earlier. If you ever wanted to submit your own data, there's instructions on how to do this here. You can read through that in detail. There's also an advanced search, but for our purpose today, we just need search box at the top and do a simple search for Blueprint. So I type in Blueprint here and hit enter. And now you can see we have actually 2 track hubs here which are called Blueprint. And the difference is that one is associated with a GRCH37 assembly and the second with a GRCH38 assembly. So I will use the second one for the GRCH38 assembly. And what I will do is I just click on View in Genome Browser and then select my most favorite genome browser, which is Ensembl. And I will be taken to the location tab in Ensembl. This takes a moment. As you will see, this track hub contains a lot of different tracks which are now imported into Ensembl. And Ensembl is loading the Configure this page menu now on the location tab. And now it's done that. And if you have a look in the menu on the left-hand side, which should look familiar to you already, there's an additional section here now on the Blueprint hub. And you can see we have 21 tracks out of 5,695, which are switched on at the moment. You can customize what you would like to see from this Blueprint hub now by going, for example, to the Blueprint region here and configuring data in a matrix, which looks a little bit ugly right now on my browser. I will skip that actually. Okay, I have closed my configure this page window now. And you can see you have additional tracks here called Blueprint Region and Blueprint Signals for a variety of cell types. I won't be discussing what this type of data is, but this is how you can import it into Ensembl. I will try the configuration one more time. So I go to configure this page. And I will use the blueprint signal this time. Which also takes a moment to load. Yeah, this looks better. So the people who have been watching the regulatory build webinar will have seen a similar matrix earlier. So we have cell types from top to bottom, and we have different types of epigenetics data from left to right, and you can select here what you want to see and how you want to display it. You can do the same for the cell types. And whatever you want to display turns blue in this matrix. And yeah, if you close that, this is how you then can customize what exactly you want to see from these 5,500 tracks. Okay. Yeah, mentioning 5,500 tracks. Again, it's important if you use with track hubs to delete them again if you don't need them anymore, and also carefully think about if you need them in the first instance, because the browser will become very slow. So to do this, you do the same as for the custom tracks. You go to configure this page, and then personal data. This hangs now. And here now you can see your data is the Blueprint Hub now. And I will delete it again by clicking on the trash bin and closing the window. And my track hub is gone. So this was the first way of getting track hub into Ensembl. You search the track hub registry and import data from there. You can also search for track hubs within Ensembl. And to do this, I will just stay on this page and visit configure this page again. And the personal data tab. On the left-hand side, you have this menu item now, track hub registry search. You can search the track hub registry from here. At the moment, I'm searching human and the GRCH38 assembly. This is because where I am at the moment in the human genome. So here I will just type blueprint into my search box again and click search. And it has found this hub. And on this page, I just need to say attach this hub using the button on the right. It tells me it has attached successfully. And if I now click on configure this page, it's actually loading it now, and in a moment you see it appearing in the configure this page menu again as before. Here we go. So you've got the same track hub again here, which is close this. And yeah, so this is the second way of how you can get a track hub into Ensembl, and the end of my demo. So I will go back to my presentation and just say a few things very quickly to wrap up. As you know, we have these Slack workspaces where we will continue to answer your questions, also on the exercises. All materials are on this training page. You can use the Slack channel to get help with the exercises. You can also use the solutions and email our help desk. In about 5 minutes after I finish this webinar, the next webinar will be presented by Emily Perry, who will give an introduction to the Ensembl REST API. So what's the Ensembl REST API? It's a way to offer language-agnostic programmatic access to Ensembl data, and you will get an introduction about how the REST API works and what data you can retrieve using it. And you will get a brief introduction on how you can write scripts to get the data you need from Ensembl on a larger scale. This starts in about 5 minutes. There's a lot of help and documentation available on the EBI page, on the Ensembl page. We have a YouTube and a Youku channel with videos which are either shorter videos on specific topics or longer ones, such as these webinars. You can email us, and you can join one of our public mailing lists. You can also follow us on social media. We have a Facebook account, a Twitter account, and we write blogs on ensembl.info. Please do cite us if you use the Ensembl resource, which is freely available. It's very important for our continuous funding and the provision of this resource that we get these citations. Yeah, this is the whole Ensembl team during our retreat in 2018. And I have to thank all of these people in the entire Ensembl team, as well as our funders. Our main funding comes from the Wellcome Trust, and we get a lot of co-funding from Ensembl and other funding agencies. So yeah, thank you very much.